Excitation-contraction coupling or contraction of cardiac muscle. The term excitation-contraction coupling refers to the mechanism whereby the action potential causes the myofibrils of muscle to contract. Before discussing the excitation-contraction coupling, we must know that the cardiomyocyte sarcolemma has transverse tubules or T-tubules, which are rich in ion channels and important for excitation-contraction coupling. These are T-tubules, which are highly branched invaginations of cardiomyocyte sarcolemma and has channels for ion, and also important for maintenance of resting membrane potential, action potential initiation and regulation, and signaling transduction. When the action potential passes over the cardiac muscle membrane, the action potential spreads to the interior of the cardiac muscle fiber along the membrane of T-tubules. Then it acts on sarcoplasmic reticulum to release calcium ions into the muscle sarcoplasm. So the calcium ions diffuse into the myofibrils to promote sliding of actin and myosin filaments. And as a result, it produces muscle contraction. This is a diagram that illustrates the excitation-contraction coupling process. As I mentioned earlier, when an action potential passes over the cardiac muscle membrane, the action potential spreads to the interior of the cardiac muscle fiber along the membrane of the T-tubule. Then it acts on the sarcoplasmic reticulum to release calcium ions into the muscle sarcoplasm. In another few thousandths of a second, these calcium ions diffuse into the myofibrils and catalyze the chemical reactions that promote sliding of the actin and myosin filaments along one another, which produce the muscle contraction. Here you can see how contraction occurs between the actin and myosin filaments. This mechanism of excitation-contraction coupling is the same as that for the skeletal muscle. But there is a second effect also. In addition to the calcium ions that are released into the sarcoplasm from the sarcoplasmic reticulum, calcium ions also diffuse into the sarcoplasm from the T-tubules at the time of action potential. Without the calcium from the T-tubules, the strength of cardiac muscle contraction would be reduced considerably because the sarcoplasmic reticulum of cardiac muscle is less well developed than that of skeletal muscle and does not store enough calcium to provide full contraction. The T-tubules in cardiac muscle are 5 times wider in diameter and 25 times larger in volume compared to those in skeletal muscle. Also inside the T-tubules is a large quantity of mucopolysaccharides that are electronegatively charged and bind an abundant store of calcium ions which are available in action potential. The strength of contraction of cardiac muscle depends to a great extent on the concentration of calcium ions in the ECF or extracellular fluid. But the skeletal muscle contraction is caused almost entirely by calcium ions released from the sarcoplasmic reticulum. At the end of the plateau, calcium ions are rapidly pumped back out of the muscle fibers. Transport of calcium back into the sarcoplasmic reticulum is achieved with the help of a calcium adenosine triphosphatase or ATPase pump, also known as SERCA2 or sarcoplasmic endoplasmic reticulum calcium ATPase, which use ATP. Calcium ions are also removed from the cell by a sodium-calcium exchanger, which does not use ATP. The sodium that enters the cell during this exchange is then transported out of the cell by the sodium-potassium ATPase pump. It also uses ATP. As a result, the contraction ceases until a new action potential comes along. Duration of contraction. Cardiac muscle begins to contract a few milliseconds after the action potential begins, and continue to contract until a few milliseconds after the action potential ends. Therefore, the duration of contraction of cardiac muscle fiber is mainly a function of the duration of the action potential, about 0.2 seconds in atrial muscle and 0.3 seconds in ventricular muscle.
Thank you for staying until the end. I will see you in the next video.